Good morning. I am going to be talking through this slideshow. You also have the option of just clicking on it without watching my recording. My recording is just meant if you need a little bit of extra information instead of just clicking through it on your own and trying to extract the information that I want you to get. So the learning targets for this week, we're going to start learning about protein synthesis. And the learning targets are, I can describe differences in the structures and functions of DNA and RNA. And the second one is I can explain the process of protein synthesis. Bear with me with this screencast. I've just learned this software and I'm, figuring it out as I click through it. So to get ready to go through this, you want to print the notes guide on Google Classroom, and then you want to fill it out as we go through this slideshow. If for some reason you can't print the notes guide, it's totally fine. You'll get a blank sheet of paper and you'll take notes. How are you going to know what to take notes of? If you look over here on the screen side, whenever you see the yellow box below, you are going to need to jot down a few notes. And you want to just read each slide, take your time to understand the concepts. All right, so we have been studying DNA, we've been studying DNA replication, and we've studied genetics. And so we're trying to understand, how does the DNA code create our traits? So if these are our nucleotides in DNA, how do we go from this to give us a trait like dimples or like bent pinkies or a widow's peak or something like that, our eye color? So that's what we're trying to understand. So the way that this happens is by making proteins. Proteins are biomolecules. We learned about them. We learned about the 4 biomolecules. And we know that the monomers are amino acids that are linked together. So here I have the protein human insulin. Off to the right, I've got one purple chain of amino acids linked together. And that purple chain is connected to this dark blue chain, which is another chain of amino acids making up the protein insulin. All right, so you're looking at the notes that you've printed, and if you have not had a chance to do that, you're just going to jot down a few notes because we see this yellow rectangular box. So on your notes, it says organelle A. What is the name of this organelle? So if this is a cell, we know what that organelle is. Question 2 is, what important molecule is found inside this organelle? So let's go ahead and take a look at that. So we know that that is the nucleus, and we know the important organelle is DNA that's inside. And we know that DNA are the instructions to build us. So if we go back to the second part, organelle B, what's the name of organelle B? Pointing to these little dots, or it could be pointing to these larger green dots. What's its function? We're looking at those two questions. So questions 3 and 4. We know that either these little green dots or this larger green structure are ribosomes. We know that they're in the cytoplasm, they're outside of the nucleus, and we know their job is to make proteins. Question 5, one thing that I want you to remember is, what are the monomers of proteins? We just mentioned this. We know the monomers of proteins are amino acids. So if we know all of this, let's start trying to understand, how do the ribosomes get the instructions? If the instructions are right here, the DNA in the nucleus, how do those instructions get out to the cytoplasm, to the ribosomes, to allow protein synthesis to happen? So if we take a look, step 1, a messenger is needed to take the information from the DNA to the ribosome. So the DNA can't leave the nucleus, and so the body has come up with a way to send a messenger outside of the nucleus to look for the ribosome. Step 2 is that message that was made is decoded, and then it's made into proteins. So again, you've got your yellow rectangle, so you're taking notes. We're going to look at protein synthesis, and there's 2 steps that we're going to look at. So we start off with DNA in the nucleus, and we have to make a message. To make the message is RNA, and it's a specific type of RNA. This process where we go from DNA to RNA is called transcription. The second step to help us make a protein goes from RNA to the actual protein, and that's called translation. Here's a little bit of a visual overview of protein synthesis. So here we have our DNA in the nucleus. Transcription occurs in the nucleus where we make a copy of the DNA. That copy is right here, and it's called mRNA. The M stands for messenger. The mRNA is able to leave the nucleus, unlike the DNA, and it's found a ribosome in the cytoplasm. It's attached to the ribosome, and now it's starting to make these little circular structures, which are amino acids, and they're getting linked together. So that's a visual picture of protein synthesis. So now, all right, so the first step that we're going to do in our notes is we're going to compare DNA to RNA. And the way we're going to do that is we're going to look at these pictures here. And we're going to try to determine, okay, DNA versus RNA, how many strands does each have? Based on what we see in the pictures, what are the letters of the nitrogen bases? You already know this, so you can already fill this in. What's the type of sugar present in DNA versus RNA? And what are the base pairing rules? So you can already start jotting some stuff down. But we can also look a little bit closer to this. So if we look at DNA, we know it's a double helix. So if it's a double helix, we know there are 1 and 2 strands. If we come over here and we look at RNA, RNA is made up of only 1 strand. If we look at the types of bases present, we know in DNA we have A and T and C and G. Or we could say T and A and G and C, but we know we have 4 bases. In RNA, we have 4 bases also, but I've circled this one here and it's in red. So we have A, we have C, we have G, and we don't have T. Instead, there's the base called uracil, which is what the U stands for. Now the next thing, the type of sugar, if we just look at this picture here, in DNA we already know is deoxyribose. Here's my sugar phosphate backbone. In RNA, it's a little bit different. It's called ribose, so it's not the deoxy where 1 oxygen has been removed from the structure. Now base pairing rules, if we look at this picture, we know that A goes with T and C goes with G in DNA. In RNA, we don't know that yet, because we don't have another strand of RNA that is present. So we don't know exactly how this works in RNA, but I provided it for you in your table. So we're going to go ahead and take a look. So we know there's 2 strands of DNA. There's 1 strand of RNA. We know the letters of the nitrogen bases are A for adenine, T for thymine, C for cytosine, and G for guanine. In RNA, we have adenine, A for adenine, U for uracil, C for cytosine, and G for guanine. The sugar is deoxyribose in DNA, and it's ribose in RNA. The base pairing rules are A pairs with T, and this says over here C pairs with G. A pairs with U, and C pairs with G in RNA. Pause this at any time if I'm going too fast. All right, this video is optional, and I'm also posting the slideshow without my voice, so you can easily go here if it will be beneficial to you to watch this video, recapping the differences between DNA and RNA. And so I won't play the videos in this talk. You can always go to the slideshow, which does not have my voice, and you can click on the videos and watch them there. And there are a handful of videos that I definitely want you to watch in here, which provide a bit of an overview of the process. So definitely don't skip them, go watch them. Because by the end of the week, I definitely want you having a pretty good understanding of this. All right, so moving on. So protein synthesis, we know we go from DNA to protein, and we know we have an intermediate step. We really go DNA to the messenger, which goes out to the cytoplasm, and then in the cytoplasm, at the ribosomes, a protein is made. So I want you to either draw this table if you didn't print the notes, or fill it in based on the information here. So transcription, it starts with what? Transcription starts with DNA. We go from DNA to mRNA in the process of transcription. It ends with mRNA, and the location of where transcription occurs is in the nucleus. Translation, if we come over here, we've got translation. We go from mRNA to a protein. So it starts with mRNA, and it ends with, we actually first start taking one amino acid, adding it to another, and to another, and to another to make the protein. So the end ultimate result is a protein, but we're linking amino acids together. The location of translation is in the cytoplasm because we know that's where the ribosomes are. All right, so you're going to jot down a few of the steps, and this is simplified, and this is just a very generalized look at transcription steps. So in step 1, if here's my DNA, I have the enzyme RNA polymerase, which is this big yellow structure here. It recognizes the start of a gene. So let's say I need to make a new fingernail, and I need to open up the gene that's going to make the protein that's going to help build my fingernails, for example. And so let's say that gene is right here, and that's the starting point. So the RNA polymerase recognizes the start of that gene, and it starts to unwind that segment of DNA. Step 2. The RNA polymerase uses one strand of DNA. This sounds familiar, right? This kind of sounds like DNA replication. So it uses one side, so it happens to be this side. I have my nucleotides that are free floating, and they're coming in and they're base pairing to the DNA here. And RNA polymerase moves along the DNA until that whole message has been made of that entire fingernail gene, that one portion of the DNA. Step 3, once that gene has been transcribed, the completed mRNA strand separates from the DNA, and it's now called a transcript. So a transcript is like a copy of something. It is able to leave the nucleus, and it's going to go to the cytoplasm. So let's practice transcription just a little bit before we talk about translation. So if this is our strand of DNA, and as you recall, the bases present in RNA are A, U, C, and G. We don't have a T. So we're going to make the messenger RNA on the bottom here, the complementary RNA strand, and there are no T's in it. So if there's a T, RNA has these 4 nucleotides. I know an A goes with the T. If there is an A in DNA, that means the mRNA is going to have a U. C goes with G, G goes with C, A goes with U, T with A, C with G. I have 3 U's in a row, C, A, U, G, and A. Okay, so make sure that makes sense to you. So this is my mRNA transcript that just got transcribed. Okay, again, I definitely want you to watch this video. This video summarizes the process of transcription, and it's a good visual. So I want you to go to the slideshow, and I want you to play it. All right, now we're going to look at translation steps. So the definition. Translation is the process of decoding the mRNA, the message, into a polypeptide chain. And a polypeptide chain is simply a chain that has one amino acid hooked to the next one, hooked to the next one, hooked to the next one. So step 1, mRNA travels to a ribosome. Here's my pink mRNA transcript. This whole brown tannish structure is the ribosome. So it travels to the ribosome. In step 2, in the ribosome, something called a tRNA, and that's this structure right here, and T stands for transfer. This reads this mRNA codon, and if there's a star right here, And an mRNA codon is made up of 3 nucleotides in a row. So I have an A, a U, and a G. So this transfer RNA reads this AUG and it brings in the matching amino acid that corresponds with this sequence. So the mRNA codon is a new vocab term, and so the genetic code, if this is my mRNA transcript, it's read 3 letters at a time, so that each word is 3 bases. For example, AUG is 1 codon, and that is going to correspond to 1 amino acid. My second codon is AAC. That's going to correspond to a second amino acid. So again, 1 codon is made up of 3 nucleotides on the messenger RNA. So you're going to practice here, and I just want you to look at the screen. And here's my DNA. This side of my DNA, my purple strip here, was used to make the complementary mRNA transcript. So let's make sure this base pairing looks good. So with the T, I have an A. C, I have a G. G, I have a C. My A, I have a U. My C, I have a G. T, I have an A. G, I have a C. G, I have a C. A goes with U. T goes with A. G goes with C. Or excuse me, C goes with G. G goes with C, and so on. So it all makes sense based on what we've just learned. So how many codons are in this mRNA transcript is the question. So remember, 3 letters on the mRNA transcript are equal to 1 codon. So pause it. Pause the video and count how many codons there would be. And then I'll show you the answer, okay? So we've got 1, 2, 3, 4, 5, 6. So if you said there are 6 codons, you are absolutely right. All right, so now we are on our 3rd page of notes, transfer RNA. That's this structure here, this pinkish structure. So the job of tRNA is to translate the message, so the message is this purple color mRNA, into proteins. So let's look at the picture of tRNA and fill in the blank and answer the below question. So what is the name of the 3-letter code at the bottom of the tRNA? So here's my tRNA. I know on mRNA it's called a codon. On tRNA it's called an anticodon. The anticodon recognizes the blank on the mRNA. So this anticodon right here, UAC, recognizes the codon on the mRNA. And if you notice, they're complementarily base pairing right there. What monomer is attached at the top of the tRNA? So methionine is an amino acid which corresponds with AUG. And so this transfer RNA knows, oh look, it's an AUG. I need to bring in a methionine. So it's an amino acid. So, and I know an amino acid is the monomer, right? We've said this about 3 times, maybe more. It's a monomer of what biomolecule? We know it's the monomer of a protein. And it's cut off my screen, but you just heard me say it. And we've said it a few times throughout the slideshow. All right, so definitely want you to watch this video. And so make sure that you go watch it on the slideshow that doesn't have my recording. All right, so here's an overview of translation. This is the mRNA transcript, okay? This structure right here, the yellow structure is a ribosome. These cross-looking structures, upside-down cross-looking structures, are the tRNAs. The anticodon on the tRNA is recognizing the codon on the mRNA. And you'll notice this polypeptide chain, right? These brightly colored circles keeps getting longer and longer. All right. So how do we figure out what amino acid goes with what codon? Well, we need a codon wheel or a table. So codon wheel, this happens to be the wheel, they list the mRNA codon that codes for each amino acid. So we're going to get a little practice reading it on the next few slides. So what amino acid is coded for by the codon GGG? So How do I read this? I start off with the inner circle and I find G. My second letter corresponds with that second circle, and it's G and it's G, and then my third letter is a G. So I go G, G, that gives me GLY, because when I go G, 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 my first three wheels, parts of the wheel, it corresponds to GLY, glycine. All right, so now what amino acid is coded for by the codon UCA? U is my first letter. I start off on this first inner circle. My second letter is C. I go to my second circle. My third letter is an A. I go to my third circle and I get SER, or serine. Pause to make sure you understand this. So what amino acid is coded for by the codon CAU? Now instead of using a wheel, I'm going to use a table. So you'll notice here this says first letter, second letter, and third letter. So my first letter is a C, so I'm coming in this square right here. My second letter corresponds with this A. So I'm in the C row and I come over to the A column. And then it's a U. So CAU corresponds to the amino acid HIS, which is histidine. What amino acid is coded for by the codon GCA? So again, G is my first letter. I come over here, here's my G. My second letter is a C. Here's my C. I come down here and it's GCA. If I look here, GCA corresponds to ALA, which is alanine. All right, so if that was confusing, go back and watch that part of the video again, or watch this optional video. All right, so you have a little bit more practice. This is on your note sheet. You have the chart right in front of you. So I want you to take a look at your chart. This chart is in your notes. And I just want you to, in your head, jot down somewhere, what is CAU? Just get a little bit more practice. What does AAC, what amino acid does that correspond to? And what amino acid does GUC correspond to? What about AGC? So if we look at this, my C is right here. Here's my A. And my third base is over here. So I have my C, I have my A, and I have my U. That's histidine. AAC, here's my first base. Here's my second base, and then here's my third base. That corresponds to asparagine. So double check these, take a look, and make sure they make sense to you. All right, so you're looking at your note sheet. What is the start codon? We know the start codon is right here where it says start, and it corresponds to the first base, which is an A, the second base, which is a U, the third base, which is a G. What codons code to stop translation? I have three words that say stop. So my stop codons are U, A, U, A, G, and U, G, A. Pause this and make sure this makes sense to you. List the amino acid sequence for the mRNA message. So if I have AUG, what amino acid does that correspond to? AUG corresponds to methionine. CAC, histidine. UCA, serine. UAA, stop. Make sure that makes sense. Pause it. Convince yourself that's true. On your note sheet, go ahead and do this problem here. So if I have DNA, transcribe the mRNA strand and then translate it to the amino acid sequence. Pause the video. I'll go over the answers momentarily. Definitely want you to watch this video, Overview DNA to Protein. And then this is an animation which goes through protein synthesis. Make sure this makes sense also, and it's got the 2 steps. So Step 1 is transcription. RNA polymerase is unzipping the DNA. I'm making an mRNA transcript. Step 2, that mRNA went and found this ribosome. The codons are getting attached to by the tRNAs.